So welcome. I know people are joining to our CTSI Translational Tuesdays. We'll wait to see others joining shortly. Appreciate everybody's work on all of these special webinars that we've started. As you know, the CTSI has started this whole new webinar on Tuesdays, and these webinars are all focusing on ongoing research and innovations for COVID-19. Happy to say this is the 6th one that we're doing. My name is Ralph Sacco. I'm the director of the CTSI. So before we get started, there are a few things to remember. First, the webinar is being recorded. Second, the Q&A bar at the bottom or wherever it's located on your Zoom menu is a way to put in questions. We will try to deal with questions at the end. And we have a short poll. Last time I forgot the poll. So please stay on to answer the poll in terms of how we're doing. Next slide. So I hope you all are aware of the CTSI resources, the resources that we've continued pre-COVID, and we are continuing. We're continuing many of our approaches to consultative services, a lot of it being done virtually. There's a special section of the CTSI website now that is all devoted to coronavirus. All of the 5 Translational Tuesday recordings are there. Links to our discussion forums that are up and running to get people working together on interdisciplinary science. CTSI supported COVID-19 research that we featured in prior webinars, and other services both for researchers in COVID work as well as non-COVID work that the CTSI continues to support for translational research. There are also some links regarding funding opportunities from NCATS, you know, the NIH Institute that funds all our CTSIs. And tune in because there's going to be some new stuff coming out regarding a national cohort collaborative that the CTSIs across the United States are going to be focused on to bring a lot of COVID data together as part of a huge data network. But today, next slide, it's really my pleasure to introduce our CTSI Translational Tuesday speaker and to focus on vaccine development. We've all heard about it, we've been reading about it in the news, and we're going to hear about the development of a heat shock protein vaccine for COVID-19. Dr. Natasa Strbo, who is an assistant professor in the Department of Microbiology and Immunology. She's recipient of Department of Defense and Florida Department of Health awards for malaria and Zika vaccine development, and is a Miami CTSI KL2 graduate. So we're really happy to have her here. And she's going to give an overview of both the efforts that are out there in developing to work on a vaccine and some of the stunning timelines that you'll see for vaccine development for the 2019 novel coronavirus. Go a little bit over the interesting aspects of this virus, including the development of obviously a cell-based heat shock protein vaccine. She's an expert in this area. You're going to hear about it, and we're really happy to welcome Natasa for this special Translational Tuesday webinar. So you can share your screen, and I will disappear.
Thank you. First, I want to thank you, Dr. Sacco, and the CTSI leadership for the kind invitation to present my COVID-19 vaccine work on these Translational Tuesday webinars, as well as to all of you joining us this afternoon. It is such an honor and pleasure to give this webinar after all previous experts and a true first-line heroes who are fighting the COVID-19 here at the UN. So thank you all. So without further ado, let's begin. So these are my disclosures. And at the very beginning, I want to acknowledge my lab members, and they're silent COVID-19 warriors that are working in the lab since day one of the stay-at-home order and doing their best to move this vaccine development project forward. So big shout out goes to graduate student Caitlin, to research associate Laura and Eva. Thank you so much. So in my talk today, I will first give a very brief overview of SARS-CoV-2 and COVID-19 pathogenicity and immune responses. And then I will explain the principle of the vaccine platforms of current front-runner vaccine candidates. And I will also talk about advantages and disadvantages of each vaccine platform to kind of set the stage and introduce the vaccine platform that was developed here at the Department of Microbiology and Immunology by Dr. Eckhard Podak at UM 22 years ago. And today we are using this platform for generation of COVID-19 vaccine. I'm very excited to share with you today a progress that we are making with the heat shock protein COVID-19 vaccine development. And of course, on the end, I will try to answer this million-dollar question, can we have a safe vaccine in 18 months? So many public health officials have expressed concern that even after the current outbreak of the COVID-19 can come back. So vaccines are absolutely the best way to ensure that doesn't happen. So how are we going to get to the safe COVID-19 vaccine? First question is, can we make a vaccine against COVID-19? And if we do, which vaccine platform will induce the protective immunity? And most importantly, how long that immunity is going to last? So we have a lot of questions and not many answers yet. But for sure, we know that the vaccine development will not follow a Hollywood movie outbreak in which Dustin Hoffman develops the vaccine within the hours of the virus discovery, and moreover, the vaccine is 100% effective. But on the other end, we also know that that we won't follow this classical vaccine developmental pathway and paradigm, where basically we have to, it requires multiple years, on average 20 years timeline from discovery to the vaccine license. So it's a truly impressive to see other new emerging disease vaccine development timeline. So if we look at the Ebola here, it took 27 years from the time from the outbreak to identification to the phase 1 clinical trial. And then on top of that, additional 20 years from the clinical trial to get the licensed vaccine. And actually, Ebola vaccine was licensed in December of 2019. Or the other extreme example is the dengue vaccine. It took over 75 years from the first clinical trial to get a licensed vaccine. And this vaccine comes with a major restriction regarding to the age as well as the previous dengue infection. And then we really have to appreciate that on the top of the list is the COVID-19 and this tiny blue bar showing the time of outbreak and identification to the phase 1 clinical trial, and it took only 3 months. So that is something that we have to have in mind when we're thinking and asking about the development of the COVID-19. This is absolutely the rocket speed development in vaccine development, and it's never seen before, and led to this 8 candidate vaccines in a clinical evaluation. And this is the data updated on May 15th. An additional 110 candidate vaccines in a preclinical evaluation. And I'm very proud to announce that our own heat shock protein-based vaccine efforts were recognized by World Health Organization and included on their list of a landscape of the candidate vaccines. And you can find the full list on this website. So like the other respiratory coronaviruses, SARS-CoV is transmitted primarily via respiratory droplets with the possible but still not proven fecal oral transmission route. So SARS-CoV-2 is the large enveloped virus with a single-stranded RNA genome, making it the largest known genome of any RNA viruses. So as a member of the beta coronavirus family, has a high genetic similarity with SARS. And it also has been confirmed that SARS-2 uses the same cellular entry receptor, and that's known as angiotensin converting enzyme 2, ACE2, as a SARS. And the virion S-glycoprotein that is shown here in green is a surface protein that latches to the ACE2 receptor to fuse with the membrane and enter in the cell. So coronaviruses, as well as an influenza, H1N1 viruses, viral entry and spread in a host is also critically dependent on another receptor, and that is the surface protease TMPRSS2. And it's also been shown that it's very important actually to activate the COVID-2 entry by enabling the protein S cleavage. So what are the cells that are expressing these receptors? So actually there is a very small percent of the cells in respiratory and intestinal tract that are expressing these two receptors. And those cells include the secretory goblet cells in the nose, and they're important for the mucus secretion, type 2 pneumocytes in the lungs, and the enterocytes in the small intestine. So it's really not many cells that can get infected with a COVID-2 and support the active replication of the virus. So as I said, the virus enter in respiratory tract, and we will really quickly go through and we'll focus only on the airway epithelial cells and the pathogenicity that COVID-2 is inducing in these cells. So upon the endocytosis and the release of the viral genome and the translation of the viral polyproteins and structural protein, replication of the genomic RNA, all that leads to assembly of the mature virions and release of the new virions. So this active replication and release of the virus cause the host cells to go a particular type of a death, and it's a highly inflammatory form of the program cells that it's known as pyroptosis. And when dead cells release a lot of molecules that are known as the damage associated molecular patterns, and that include the viral host DNA, ATP, as well as the heat shock proteins. And we're going to come back to this. And all these molecules are then recognized by these neighboring epithelial, endothelial cells and alveolar macrophages, and they are actually triggered to generate a very strong pro-inflammatory responses, which include all these cytokines we've been hearing a lot in the previous webinars, and those are highly pro-inflammatory, IL-6, MIP-1 alpha, and these cytokines then attract the T cells, monocytes, macrophages, and they are additionally then produce same cytokines and basically promoting the further inflammation and promoting this pro-inflammatory feedback loop. So many viruses develop a very refined mechanism of immune evasion in order to increase viral replication. It's exactly the same for SARS, MERS, and COVID-2. So all of these viruses have genes that interfere with innate responses and block the antiviral responses to increase the virus replication. So we really don't have a time to go into details and look at all these individual viral proteins that are involved, for example, in an inhibition of translation of type 1 interference or blocking of the recognition of the virus, as well as the SARS-2 have this effect effective suppression, and it's really important, includes this major signaling pathways of blocking NF-kappa B and a further production and activation of the type 1 interferons. So though seemingly contradictory to mechanism of immune evasion, enhanced innate immune activation, including increase of this pro-inflammatory cytokine IL-6 and TNF, for example, contributes to morbidity and mortality of the COVID-19. So one possible explanation is the induction of endothelial and vascular cell damage and the cell death as a result of this continuous viral replication and dysregulation of innate immune responses. So basically virus is inducing, as I said, apoptosis and pyroptosis, and these are, and this results in the primary inflammatory responses. And as I already mentioned, that will include this tremendous production of cytokines, the cell infiltration, increased vascular permeability. In addition, it's known now that the COVID-2 directly influences the ACE2 downregulation and shedding, and that all contributed further to the RAS dysfunction and more of the primary inflammatory responses. Recently, we also looking much more in these secondary inflammatory responses, which are a result of the virus and antibody complexes, which then induce and lead to the activation of the macrophages, further activation of the cytokines, complement system activation, antibody-dependent cell cytotoxicity will lead to the cellular damage as well as a direct cytotoxic effect of a T lymphocytes. So all this contributes to this completely dysfunctional immune responses. But we really have to focus that only 50% of the people infected with the COVID-2 have this severe clinical outcome, and 5% develop life-threatening illnesses. For the vaccine purposes, it's really important to focus on this other side of this slide, and those are on all the immune responses that are present in this mild clinical outcome, and in the people that are infected without symptoms. Why? Because in this case we have the infected cells that are rapidly cleared and recognized by the T cells. There are macrophages that recognize and phagocytose apoptotic cells that do not produce this tremendous amount of pro-inflammatory cytokines, and all the neutralizing antibodies that bind and they inactivate virus. So this is all the picture of very controlled viral infection, and that is a really most important to focus when we talk about the vaccine development. Understanding this immunological mechanism that control infection is a golden key of the development of the protective vaccine. So we are really, so right now, currently, we know little about what constituted protective immune responses against COVID-19. So we learned a lot from the SARS and from the MERS infections, and we are getting every day more and more information about recently affected COVID-2 patients. So overall, we've learned that there is a relatively high level of immune responses after infection, especially antibody responses to the surface of this S spike protein. Traditionally, we are focused more on the memory B cells and antibody responses, so there is much more information about antibody responses than about the T cell responses. However, T cell immune responses have been documented in SARS-CoV specific memory T cell responses have been found in the people that recovered from SARS even 11 years after initial infection. While, for example, there are reports of relatively fast waning of the antibody responses. So they've been found up to 2 years after the infection. So, however, the in vivo data on the type and the level of immunity required to protect from subsequent reinfection is currently unknown. So we didn't have the reinfection with MERS and SARS. So we're really missing this very, very important information. But as I said, we are getting much more knowledgeable about the antibody and T cell responses in COVID-2 patients, and more importantly about their breadth and kinetics during the COVID-19. So it was recently reported about acute antibody responses in a COVID-2 patients, actually 285 patients in this particular study. And they shown that within the 19 days after the symptom onset, 100% of the patient tested positive for antiviral immunoglobulin IgG. But what was also very interesting in this study is their report that basically titers of the COVID-2 positive of COVID-specific IgG responses were higher, significantly higher in the severe, in groups of severe cases than in the less severe. So we really don't know if this is a reason for severity, or it's just consequence of, for example, higher viral load. So this type of the studies are really necessary that we learn more about protective immune responses. And then also for the vaccine development purposes, it is equally important to study immune responses in the patients without disease or just with a mild disease. So this Australian group follow-up a 40-year-old woman admitted at day 3, what they found is a really high frequency of these antibody-specific, antibody-secreting cells and T follicular helper cells, which are really crucial for the development of the antibodies, as well as this very fast peak in the CD8 T cells, activated CD8 T cells that have a lot of cytotoxic molecules as the granzymes and perforin, and really not much happening with the NK cell and monocytes. They didn't find pro-inflammatory cytokines. So overall, this was the first study that provides a novel contribution to understanding of the breadth and kinetics of immune responses during the non-severe COVID-19. And furthermore, this study really indicates that this robust multifactorial immune responses can be elicited to COVID-2, and also that early adaptive immune responses may correlate with the better clinical outcome. Of course, we do need a much larger cohort of people with the COVID-19 with different disease severities to determine whether they could be used and predict disease outcome and evaluate new interventions that may minimize the severity of disease. But something that I want to point out during this talk that it's really, I think, very interesting regarding the T cell response during the COVID-19 infection. So COVID-2 specific T cells were found in all of the convalescent COVID-19 patients. But what it's really getting a lot of attention these days is this recent finding, actually published this week, that SARS-CoV-2 reactive CD4 T cells were found in 40 to 60% of unexposed individuals. So these are samples that were collected between the 2015 and 18. So they were not in the contact with a COVID-2, but that really suggests this cross-reactive T cell recognition between circulating common cold coronaviruses and SARS-CoV-2. So why is this important? Because cross-reactive immunity, for example, to influenza strain has been modeled to be a critical influencer of susceptibility to new emerging and potentially pandemic influenza strains. So we really have to pay more attention to this data and definitely look at it in a larger cohort. So from all this, the question is, so, okay, with all this knowledge, how are we going to develop a protective vaccine? So we current knowledge agrees that we should focus on the antigen presenting cells. Why to the antigen presenting cells? Because they do play a key role between the innate and adaptive responses, and they're critical to determining the direction of the adaptive immune responses. So this really ideal vaccine should initiate the innate immune responses that is capable of directing adaptive immune responses toward efficient activation of the helper CD4 T cells that will promote the B cells and absolutely highly neutralizing anti-coronavirus antibodies, and in the same time help give help to the cytotoxic T cells that can destroy infected cells. And then the bottom line, they all have to induce a long-lived a memory B and T cells. So how are we going to achieve that? So general rule is that the more similar vaccine is to the disease-causing form of the organism, the better the immune response are going to be developed. But in that, with knowing that rule, we really need to think about balancing immunogenicity and tolerability. That it's really, really crucial because in a case of these purified antigens or well-known recombinant proteins, they generally have a very improved tolerability and safety profiles compared to the live attenuated pathogens or inactivated pathogens. But at the same time, there are also much less immunogenic due to the removal of these pathogenic features of the organism. So the one way we can improve this immunogenicity of the purified antigens that we used in a vaccine is by adding immune stimulants such as adjuvants, and we're going to mention those a little bit later. Or we can simply use a vaccine that is self-adjuvanted vaccine, so it doesn't need the adjuvant, and that's something that it's really a big difference of our heat shock protein. Another thing that it's really important is the choice of the immunogen. So pathogen proteins have a different level of immunogenicity, and our detailed analysis and structure of the conformation is needed before we make a decision that this particular protein has to be included in a vaccine. So far, the highest level of immunogenicity was described for the surface protein S, for this glycoprotein, spike glycoprotein. So the majority of the vaccine platforms incorporate this protein as a major immunogen. The array of the current vaccine is really huge. So you can see that there are the platforms that use the virus, so viral vector, nucleic acid, or protein-based. And majority of the approaches are based on a viral vector or on a protein base, which are definitely some very traditional vaccine approaches and kind of safe way to go. I will briefly discuss these vaccine platforms that are currently in the most advanced clinical stages, so phase 2 and phase 1, that includes the inactivated viral vaccine, non-replicating, as well as the RNA-based, which we all know is getting a lot of attention, particularly yesterday and today, with the entering in the phase 2. So this is a little busy slide, but I was trying to put together all the traditional COVID-19 vaccine platforms and their representatives. So we all know that one of the oldest and very effective vaccines, such as measles, mumps, and rubella, is the attenuated viral vaccine, and also polio as a major example of inactivated virus. So when it comes to the COVID, CoronaVac is the vaccine that is further along in the development, and it's basically the inactivated version of the SARS-CoV virus in combination with the well-known adjuvant alum. This vaccine predominantly induces neutralizing antibodies, and protective efficacy has been confirmed in the COVID NHP infection model, and just a few weeks ago was published in Science. This vaccine is also the most controversial vaccine because some previous inactivated virus vaccine candidates for SARS have been associated with a higher vaccine risk for the pathogenicity in a different animal models, and we'll come back to towards the end on that. When we look for the viral vector vaccine, we have the replicating and non-replicating vectors, and in particular in these non-replicating vectors, the backbone of the vaccine is non-pathogenic viruses, such as, for example, adenoviruses. In the case of the COVID-19 vaccine, this coronavirus spike gene is introduced into the backbone of the virus, allowing the expression of the protein S and induction of specific immune responses in the host. The vaccine that is further along and is also getting a lot of media attention is coming from Oxford University, and includes chimpanzees adenovirus, CHAD OX1. This vaccine candidate comes with an excellent vaccine record from the previous vaccine candidates based on the CHAD, on the chimpanzees adenovirus, and those are MERS, influenza, Zika, and chikungunya. So this vaccine also predominantly induces neutralizing antibody responses, but also some T-cell responses. Currently, efficacy studies have been performed with MERS candidate vaccines and non-human primate models, and showing a really good results as well. Looking at the recombinant protein and protein vaccines, using basically two major approaches, either just a receptor binding domain of the protein S, and Walter Reed is pursuing this vaccine platform, while, for example, some other are really focusing on the trimeric S protein and doing the vaccine preclinical studies with this form of the protein S. Both this approach is traditionally inducing exclusively neutralizing antibody, and what we also have to say that they really depend on the adjuvant that is used in combination with these recombinant proteins. And then, of course, the most attention are getting the more novel types of the vaccine platform based on the nucleic acids. So that's DNA and RNA-based vaccines. So we have here in the United States, we have Inovio that is developing a DNA vaccine, and Moderna RNA vaccine. So my major difference from traditional, this virus and viral vector or recombinant protein vaccine, is that DNA and RNA express the coronavirus genes, in this case the protein S gene, and they first needs to be translated to the protein and inside of the host cell, and then that protein, of course, is delivered to the antigen presenting cells for activation of immune responses. So both of these approaches are particularly DNA approach also requires additional adjuvants to enhance the immunogenicity property of the vaccine. But when it comes to the immune responses that are induced by these vaccines, it's a major difference with all of the ones that I talked before. So this one induces a T and the B cell, so which is really really important for the final outcome. So which vaccine platform is the best? So all of them have some advantages and disadvantages. We'll really quickly go through this. Attenuated and inactivated viral vaccines are definitely one of the oldest and very traditional vaccine approaches. There are straightforward processes. We have a lot of existing infrastructure that can be used, and it's no wonder that one of the current candidates that are further along in development are exactly from this group. But and they were tested in humans for SARS. There are adjuvants that have been used with this vaccine platform, but there are definitely a lot of disadvantages. First of all, the safety concern. As I said, the previous candidates did show the vaccine enhanced pathogenicity. So safety testing is the must for these type of the vaccine. And there's always, you know, the cautious and with the production of a large amount of infectious virus and also concern about the antigen and epitope integrity during the inactivation that could occur during the activation. When we look at the viral vector vaccine, the biggest advantage is there is no infectious virus. Basically, they're using a non-pathogenic virus as a backbone, and they do have excellent preclinical and clinical data. Primarily, Ebola, for example, is recombinant, is a viral vector vaccine, and there is the biggest disadvantage is a vector immunity, and that could really negatively impact the vaccine efficacy. For the DNA RNA vaccines, the huge advantage is as well no infectious virus. It's easy to scale up in the rapid production. There have been tests done with the SARS. And they look pretty good, and it's kind of similar for DNA and RNA. But I will mention that these are the most novel vaccine platforms, and we really don't have a lot of clinical studies done, and that why we really have to be still cautious. And there are a lot of things that we will learn from these platforms, and that's why the safety studies are very important for these novel vaccines. And for the protein vaccines, as I said, these are very new ones. They're traditional, and the adjuvants that are used with these vaccines are very important for the outcome. So can we do better? Can we think outside of the box? And I said yes. Absolutely yes. We developed a cell-based heat shock protein vaccine, and what is the major advantage of this vaccine platform? There is no infectious virus, but what's very important that closely mimics a native viral infection. As I mentioned before, heat shock proteins are part of the normal response to the viral infection, and that leads to the T cell responses. If we compare the heat shock protein to all the platforms on the market, it's a quite newer platform. It's an engineered cell vaccine, and it has been used in a wide range of the cancer treatments, as well as in infectious diseases as HIV, malaria, and Zika. Very important is that it induces this antigen-specific CD8 T cells, and I'll talk about it just in a minute, about the importance of induction, this multi-epitope specific T cell responses, particularly at the site of infection. And in this case, those are epithelial tissues in lungs. Today, to Heat Biologics, so it's a pharmaceutical company that we are collaborating, has completed enrollment of the phase 2 clinical trial using the GP96 to treat non-small cell lung cancer. And actually, it has in all combined clinical trials exposure of over 100 patients with an excellent safety record. So for all of those who are wondering, so what are the heat shock proteins? So heat shock proteins are these natural proteins expressed in all of cells in our body. And their main function is to chaperone the proteins and make sure that the proteins are folding properly. However, it turns out that the heat shock proteins are also these danger-associated molecular patterns that I mentioned before, and they are released from a dead cells. So once the GP96 is outside of the cell, it serves as an adjuvant. And that adjuvant activates the antigen presenting cells through the TLRs, and very importantly also engage this particular receptor, CD91, which is a key receptor involved in a process called antigen cross-presentation. And this process allows the exogenous antigens, such as pathogenic antigens, to be presented to T cells, particularly CD8 T cells. So overall, the end result is activation of innate as well as adaptive immune responses. So basically, GP96 is an adjuvant for activation of the innate immune responses, antigen carrier for priming activation of antigen-specific T cells. So Dr. Podak, our late chair of the
Natasa, are you back?
Yes, I'm sorry. I just noticed. I don't know where you guys lost me.
You just said Dr. Podak, our late chair, and then we went blank. I hope there wasn't a lightning strike over there.
Yeah, I hope to. It's sunny here. Alrighty. So just a moment. Okay. So yeah, so the Dr. Podak was, as I said, late chair of the Department of Microbiology and Immunology, my mentor for many years. And in mid-90s, he engineered secreted form of GP96 simply by changing the property of this endogenous GP96, adding a parts of immunoglobulin molecule to GP96 and making this fusion protein that can be secreted outside of the cell. And basically cells just serves as a pump that continuously pumps GP96 intracellular space. So this was a true genius invention, and it was tested in many different models, as you can see here, and published in numerous peer-reviewed articles. So Dr. Podak actually expressed the GP96 therapy. He expressed this protein in the different tumor cell lines and generated a first secreted GP96 therapeutic cancer vaccine that was later then licensed to the Heat Biologics and tested in many different cancer clinical. Oh, I'm so sorry.
You're back. I don't know why this is happening, but
yeah, this is the first time.
we've been flawless for the last 5. There are thunderstorms in the area, so.
Yeah, so I'll just go. It's a power of the GP96. All right. So my lab is then really focused on the development of a different infectious disease vaccine platforms, including the SIV, HIV, malaria, and Zika. So heat shock proteins are special interest for this infectious disease approach because they are basically the peptide fingerprint of the protein expressed in particular cells. So when we combine the antigens of a different pathogens with the secreted GP96, we actually have a multi-epitope specific vaccine. We're inducing this multi-epitope specific vaccine responses. So we
sorry to interrupt. We don't see your slides.
And we generated vector, so the plasmid DNA that expresses the gene sequence of the fusion protein GP96IG, as well as the SARS-CoV-2 spike protein S, the full length. And we express this construct in the cells that are expressing then the GP96 and the spike protein. So I'm very excited to share with you this picture of the cells.
Picture, Natasa. For whatever reason, your slides aren't being shown. And this is a picture. Can you hear us?
Oh.
Yeah.
Yeah, again, I'm so sorry. This is.
And we didn't see your picture, so make sure you're sharing the screen again if we're back.
Okay. That we do have the vaccine cells that I'm very excited to share. This picture of the cells that are growing in the lab, that are producing GP96 and the COVID-2 protein S. So now we are ready to actually quantify the production and dose the vaccine cells, so we can start performing the immunogenicity experiments in the animals. So this is really exciting phase. So what we know about our vaccine is that when we inject the vaccine in the cells, then this cells that are producing GP96 and chaperone the antigens, they are inducing the dendritic cells, the particular subsets of dendritic cells that are able to prime and activate the T cells in such a way that they can get into the epithelial tissues. We have proved that this vaccine approach can induce T cells that are effective, and they contribute to the protection against the pathogenic simian immunodeficiency virus. So we collaborated with Dr. Franchini and Dr. Vaccari from the NIH, and we showed that the GP96 in combination with recombinant protein GP120 can significantly delay acquisition of the SIV virus after the mucosal infection, which you can see here. We can also show, which is very important, what we learned from this study, that the GP96 can serve as an excellent adjuvant to B cells and induce the so-called Th1 type of antibody responses and correlates very well with the virus protection. We also did a lot of gene array analysis and found that the GP96 is inducing genes involved in B cell development and the type 1 interference. And then further on also Dr. Franchini performed this additional analysis and found that the protection from mucosal SIV infection after GP96 recombinant protein vaccination was associated with particular RAS pathway and activation of hypoxia and inflammasome in the monocytes. So with all of that, I would like to summarize and show the overall principle of the GP96 COVID-19 vaccine approach. So we generated the cells that secrete GP96 and chaperones the different epitopes of the protein S. They can activate the antigen presenting cells in the host and induce induce the cross presentation of these peptides to the CD8 T cells and induce this specific COVID-2 specific T cells then can migrate to the lungs and really induce the direct cytotoxic effect on the infected epithelial cells. So this is just an overview, and we really think that GP96 approach has this a huge advantage from all of these traditional vaccine approaches when I was talking before. Not only that we don't use infectious virus, but it's really important, this self-adjuvant component of the vaccine and this multi-epitope specific T cell responses that can be induced and also can be used as additional adjuvant to the antibody responses. There are, of course, some disadvantages, which are primarily that are correlated with this impractical and for large manufacturing, but Biologics is really working hard to improve that component. So overall, we believe that pairing our GP96 vaccine technology and expertise with the industry and HEAT's regulatory and commercial capabilities, we are reinforcing our commitment to do everything we can to combat this escalating pandemic as quickly as possible. Antibody or T cells, what should be induced and generated by the vaccine? Definitely both. For the successful vaccine, we should think about both humoral and cellular immune responses. Risks are there. We don't have much time to talk about it, but everybody, and we can cover this in actually in a Q&A session. That there are known mechanisms that could induce potential risks associated with the vaccine development, and the way how we can mitigate that is really to think to make the vaccines that have this conformationally correct antigens and high quality of neutralizing antibodies. On the other side, if we think about the T cell vaccine, that should be really Th1 bias responses and the CD8 responses, just as what we're inducing with the GP96. So can we have the vaccine in 18 months? I will say yes, because following this outbreak paradigm with the overlapping the phases, and that's really tremendously shortened developmental time, and everybody's doing that, and that's why we see the results of phase 2 coming within the 3 to 4 months of the beginning of everything, and we're really trying to follow this timeline as well, so that we can produce we can have the vaccine, but there are a lot of challenges, and first of all is the phase 3. So phase 3 is a much longer, it takes a much longer for the phase 3 for the efficacy. There is some idea about the human challenge studies, a lot of ethical questions, a lot of think about manufacturing, distribution, but we have to be safe. That's a number one, and I would finish with that safety is a priority, and is no cutting corners. My acknowledgement on the end, just for Dr. Podak, that is absolutely one of the greatest minds and inventors of our century, who left a huge legacy. And of course, my girls, my silent warriors in the lab and the whole Department of Microbiology and Immunology. And always CTSI, that was a first award that I got as investigator, and it's always so close to my heart. So I will stop there.
Thank you very much, Natasa. One, for putting this all into perspective, including all the different vaccine types out there that are all trying to work globally to affect us and improve things in this pandemic. And yours is very unique, without a doubt, and hits both B and T cells, which is great. It does have some issues, and some of the questions are coming up that I'd like to try to address. First of all, have there been any other safety profiles with this GP96 approach in other, say, non-SARS-CoV-2 approaches?
So yes, as I mentioned, so we definitely, you know, in this long period of time, we performed a lot of preclinical studies, and we never run into, and we were looking, of course, of the safety part and safety of the vaccine in the small animals, and we never seen any autoimmunity or anything that, you know, we usually look at as in a safety when we're testing a vaccine, as well as a non-human primate. So we have 2 large non-human primate studies with no any autoimmunity, inflammation, and reactivity.
Human studies, though? How about the human studies?
Yeah, and the human studies as well. So all of these 180 patients. So this is just was reported as you know, inflammation, hardness on the site of the injection, okay. And actually that was correlated with the success of the outcome. You know, looking at the induction of, for example, CD8 T cells, but no major side effects were found in none of these patients.
Great. In general, people always worry about mutations, right? Mutations of this virus, even though there's been some reports that at least in some of the SARS, the mutations aren't as fast as we think. If there were mutations, how could that affect the efficacy of your vaccine compared to, say, other vaccines that are, say, antibody specific?
Yeah, so there's something that's something that I kind of try to emphasize during my talk, that the power of the GP96 is really multi-epitope covering. So that's something that when we are thinking and when we are generating vaccines and including the immunogens, really looking also to cover the parts of the protein that has a very high homology, and it's preserved within the different viruses. So that's something that really can bypass this problem of the mutations, because each of the virus has this constant regions that are same within the family, and this is something that we're really excited, especially when, you know, in this particular set, when we're talking about COVID-2, because that's a big...
Within an individual that, say, gets inoculated with the GP96, how long does it sometimes take to develop, say, immunity?
Yeah, so these are the studies that, you know, we done, as I said, in non-human primates. Mm-hmm. You know, it's always not so extensively as we wanted, but we actually we can see that, you know, this long immunity that we were measuring 6 months after the last vaccination, we can achieve after the 3 doses. Of course, we also think with the proper you know, dosing, we could probably go with the 2. So that's something that we definitely planning in our future studies when it you know comes to.
And you know, you showed about, I think it was 110 vaccines, and you broke them down into various categories, some that are a little faster along than others. In general, are there any other T-cell, more specific T-cell related vaccines out there?
So no, I mean the closest is really you know what's been shown for the RNA vaccines, that they are really capable of inducing these T-cell responses as right. well as, you know, the antibody, the B cells. There are also some viral vectors that are reports that equally induces T and B, but really, you know, when it comes to induction of the CD8 responses, we are absolutely on top of the list. We did some side-by-side studies, for example, with the ED5 vaccine, and the data is amazing. I mean, when you look at the induction in the epithelial compartment, you basically don't see anything with the ED5, and you know, you have the GP96, 20% of all the CD8s are antigen specific, so it's a huge difference.
Another great question here is, if somebody has some immunocompromise, and let's assume they don't have COVID-19, you want to prevent it, will these kind of different viral approaches in an immunocompromised person work as effectively, including yes. yours versus the others?
Yeah, that's, you know, that's a really good question, and I don't think I have an answer for that. We have to look at the, you know, look really in the details. What we know that, you know, it's really important what kind of immune responses are you inducing. So that, you know, being such a strong Th1 bias vaccine, we highly, so we didn't test that yet, but we do have, you know, our hypothesis that that it will work in the immunosuppressive as well, just because this innate component of the vaccine and powerful innate immune responses that you're inducing. So we think that could be bypassed with this.
And a lot of questions about, you know, again, timing. If you did say your human studies work out, the manufacturing, you implied that it takes a longer time. Can you give us some handle on like how long, both for manufacturing to really put this into mass production?
Yeah, so I mean, I can you know, talk about the GP96, not so much about the other approaches, but that is you know, something that is a bottleneck right now. It is, you know, a limited cGMP facilities, and you know, these are cells that have to be cultured, and that is really, you know, the timeline and the level and robotics. So this is one of the things that we definitely would like to test and go really more automated systems, as like I know the Lonza is having this ability, so that could definitely you know, speed up the manufacturing. But idea is actually that the GP96, at least in this cell-based vaccine, could be really useful for not such a large distribution, so that everybody is getting this vaccine, but really going for the vulnerable populations. So that's something that we would really like in the future to look it up. And as well, if in elderly, we didn't talk about that, an immune response in elderly is definitely different than in any other age group. So, you know, going with this approach that could really induce a good immune response in the people 65 and over is something that, you know, our vaccine approach would be then really good.
So even if there were, say, multiple vaccines out there, there may be some that will work in large populations of younger, and then some that could be addressed in older or specific populations. You know, I think there's a lot we don't know about, you know, why, say, for example, some people may get this virus and then have a severe reaction, a much more you know, inflammatory response than others. If we can differentiate, you know, some of the innate responses that we all have, that may also select people appropriately for certain kinds of, you know, vaccine approaches.
Yeah, absolutely.
You know, and I think that goes into the whole amount of research that's going on to fit, you know, our understanding of high risk, the response to the vaccine, to the exposure. And then obviously the proper treatments. Yeah, there are other questions out there on antivirals that really wasn't the topic today. I know we're mainly talking about vaccine development. And then, you know, and I think we've covered quite a number of the topics at the moment. I know we're over time, and I do try to make sure we stick to 1 hour. We had some technical difficulties. Yes. Any last comments from you?
I would maybe just to say that, you know, when we're looking at all these candidates, vaccine candidates, it's really busy. There are 100 and over 110 candidates. It's really not a competition. It has to be, we have to learn from each other. And this is where, you know, the whole world is a big lab, and it's more than ever. It's really important that this information comes out as soon as possible, so that you can do an adaptation of the vaccine, because it's a new virus and we really don't know much about it, and you know, how we're going to fight it if we don't know what the virus does.
And that's why it's all about team science and collaboration and interdisciplinary people working together. So I know we're at the end. I want to put up the poll. I think, Daru, do you have our poll to put up? And it's up there. Did the seminar meet your expectations for this specific COVID-19 topic, which was specific to a vaccine and vaccine development? And do you plan to attend future ones? We have some other ones planned moving into the rest of May and June. I want to thank everybody, but I especially want to thank Natasa, our former KL2 awardee, and we're really proud of the terrific work you're doing carrying on with this approach, both for other work you're doing with cancer and vaccine development and for helping COVID-19. Thanks for everybody for tuning in to our CTSI Translational Tuesdays, and we hope to see you in the future in another one. Thank you.
Thank you.